Now this one second video of translation process. In the first video I discussed up to this, and in the previous is I have to discuss something. You follow that through the A site new tRNA with specific new amino acid enters to this ribosomal subunit, and from P site the tRNA moves to the A site, and from A site tRNA moves to the P site, and new tRNA enters through the A site, and from the A site moves to the P site, and from P site to the E site, and which tRNA located in the E site, this tRNA go out from the ribosomal subunit, and new tRNA enters through this A site. Now when suppose methionine containing tRNA go to the E site, at that time methionine shifted to the A site amino acid and ultimately formation of a bond. You just follow that from the P site when tRNA moves to the E site, this amino acid at that time moves to the previous amino acid or A site tRNA containing amino acid, and methionine and phenylalanine ultimately formed a bond. So peptide bond formation occurs at the A site, and now this tRNA that contains phenylalanine with methionine moves to the P site, and new tRNA come to this site, and when P site containing tRNA go to that E site, at the time methionine phenylalanine moves to the A site tRNA amino acid, and there are two peptide bond, 3 amino acid. It is simple one that when peptide bond formation occurs at the A site, and this tRNA go to the P site, and when this tRNA go to the P site and now go to the E site, that at that time this peptide bond or elongated amino acid chain moves to the A site. So suppose here 5 amino acid containing tRNA at the P site, and that tRNA go to the E site, at that time elongated amino acid chain move to the newly coming tRNA amino acid. So follow that here peptide bond formation between methionine and phenylalanine. So in the A site there is peptide bond formation and protein or amino acid chain elongated. Here you follow that this amino acid this protein or peptide bond formed between two amino acid. This was formed at the A site, and now from the A site this tRNA go to the P site. Suppose here new amino acid come to this site, and this one come to this E site. At that time this peptide bond or elongated chain of amino acid moves to the A site tRNA containing amino acid. Suppose here valine present. Valine containing tRNA come to this site, and this amino acid go to that this tRNA go to that E site. At that time this methionine phenylalanine attached with the valine containing tRNA here. Here suppose methionine, phenylalanine, lysine, arginine, and here another tRNA, new tRNA come. Suppose new tRNA come with suppose lysine, and this tRNA move to the E site. At that time this whole chain of the amino acid moves to the A site of the lysine containing tRNA, and elongation in the A site occur. So elongation at the A site and shift from the P site to the A site of this amino acid chain. So ultimately this one is protein, specific protein. and when at the initiating stage that AUG present, and at this position initiation occur, and ribosomal subunit moves to this site step by step. Here was initiation complex. Now this initiation complex come to the site, and when it find out that this stop codon UAA, there are others stop codon also. You will find from lecture class or ABC book. There is stop codon is UAA. This one is when is this stop codon is scanned by this ribosomal subunit, then at the time termination, this is called termination codon or step codon. At the time releasing factor come to this site and causes separation of this small ribosomal subunit, large ribosomal subunit, tRNA, as well as cutting this or separation of this amino acid chain from this tRNA. Here you follow that small ribosomal subunit is separated, large ribosomal subunit separated by this releasing factor. At this time releasing factor acts. Releasing factor causes separation of this two ribosomal subunit tRNA and separation of the elongated chain of amino acid from the tRNA. And tRNA and ribosomal subunit is recycling of this tRNA and ribosomal subunit, and this specific protein is synthesized through this pathway. This is the process of translation. Now I will teach you something about antibiotic, how antibiotic acts through the translation process. Suppose there is you will find some antibiotic activities like erythromycin. This is diphtherial toxin. Cat can cause this diphtheria. So erythromycin antibiotic, diphtheria toxin, how effects. Suppose erythromycin antibiotic we intake. This erythromycin causes irreversibly to binds irreversibly to this step and block this the tunnel by which peptide leaves the ribosome and thereby inhibiting translocation. Here just this step is inhibited by this erythromycin. So antibiotic, suppose this translation process occurring in the bacteria in our body, by taking this erythromycin antibiotic, we can inhibit translation process or protein synthesis process at this five number step of the bacteria. So translation process is inhibited. Ultimately bacteria cannot produce protein and ultimately it can die. Then diphtheria toxin also inhibit this process in our body. That's why we become sick as desired protein cannot be synthesized due to diphtherial toxin inhibit this step of the translation in our body. And by the erythromycin we inhibit translation process at the 5 number step of bacteria, and diphtheria toxin inhibit in our body of our replication process, translation process at the 5 number step. There are some antibiotic also, chloramphenicol. Chloramphenicol acts at this step where peptidyl transferase or peptide bond formation occur by this enzyme. This steps is inhibited by chloramphenicol antibiotic within the bacteria. If we intake chloramphenicol, this step is inhibited by the chloramphenicol within the bacteria and peptide bond cannot be formed. Here you follow that inhibition of this prokaryotic peptidyl transferase enzyme. Here puromycin antibiotic also acts through this pathway. So if we discuss that how antibiotic acts through this pathway, that inhibition of translation process or protein synthesis process in different step of bacteria is done by this one is widely used, tetracycline. Tetracycline acts interact with 30S ribosomal subunit blocking access of amino acid tRNA at the A site. It means at the A site, at this step, at the A site, inhibition of tRNA entry in the bacteria translation process. If we block the A site, new tRNA cannot enter through the A site. That's why by tetracycline intaking, we can inhibit a site of the bacteria, and bacteria cannot elongate the translation process. As a site is blocked, new tRNA cannot come. So different antibiotic acts through different pathway, and ultimately translation process of bacteria is inhibited at the different step by taking antibiotic. So up to this translation I discussed. Here ribosomal subunit or initiation complex move forward. mRNA is fixed. mRNA does not move. So at the initiating step there was AUG, and at the last time step by step it moves toward and stop codon come. So translation is completed. Thank you.